Day 5 of explaining something very simply under 2 minutes so I can understand it better. So I spoke about cancer heterogeneity and the different types. I spoke about genetic yesterday, and today I'm supposed to talk about epigenetic heterogeneity, but I realized before that I should explain what epigenetics is first. So, genetics is like the DNA code that is there in every cell, and epigenetics is what decides which part of that code will be active or inactive. So think of it like this. There's a book, and every single word, line, sentence that's written in that book is DNA. And now you take a pencil and you underline certain lines in words and you cross out certain lines in words. That is epigenetics. You read the underlined parts more often and you do not read the crossed out parts at all. So there are a few interesting examples. There are two twins, and twins have almost identical DNA, but one of them develops a disease and the other does not. Why does that happen? Because of epigenetics. The environment can influence your genes and decide which gene stays active and which does not. And that is what allows one twin to have a disease and the other twin to not have the disease, even though the underlying genetics is the same, but the epigenetics is different. The second example that I'd like you to think about is, if you take DNA from a hair cell, your saliva, your blood, your liver cell, heart cell, all of these cells will have the same DNA, right? So why do they have a different structure, a different function? Why do they look so different, and why do they perform such different roles? That again is because of epigenetics. So in a cell, epigenetics decides which genes are upregulated, that means expressed more and active, and which genes are downregulated, that means expressed less, inactive. Now there are different ways that this happens. One epigenetic mechanism is called DNA methylation. So a methyl group is CH3. A methyl group going and attaching to any letter on the DNA goes and silences it. So basically that particular gene will be silenced. The second epigenetic mechanism is histone modification. So histones are proteins that the DNA wraps around. The DNA can wrap around tightly, making it difficult to read letters, to read genes, or loosely, making it easier to read the genes and express those genes more. So